All right then. In this part of the lesson, we are talking about synaptic transmission. So let's understand this picture first. So here you are looking at three types of neurons. So this is the sensory neuron, right? And then next to it, you are looking at the relay neuron. And then we have this motor neuron, this one here. Okay, so as we have discussed in the previous lesson, that the nerve impulses were actually being conducted through these saltatory conduction, actually through these as it was jumping from one node of Ranvier to the next, right? And these are all myelinated axons, right? But as it reaches this terminal axon, reaches and joins this relay neurons actually through the dendrites here. So see, these parts are not myelinated, right? So what happens here, action potentials are generated in this case through the neurotransmitters, right? It doesn't happen because of opening of voltage gated channels as it was happening here, as the voltage gated channels were opening and sodium was diffusing in and causing depolarization, right? What is happening here is that these end terminals of the axons, they contain vesicles containing neurotransmitters. Right, so as these neurotransmitters release, they actually get attached to the receptors on the postsynaptic membrane, so which will be the membrane of this dend right here, right? So they will attach to those receptors, which will change the shape, and this will actually allow the chemical gated sodium channel to open, and through that the sodium ions actually diffuse in and cause depolarization of the membrane, right? So here actually chemical gated channels will open, not the voltage gated. So this is a difference here, like here in synapses, as action potentials are being generated here in the synapses, right? Unlike here, like in myelinated sheets, that it was actually jumping from one node to the next, right? But it was actually happening because of the opening of voltage-gated sodium channels, right? So the same happens here. And similarly, as if the nerve impulse is high enough, it crosses the threshold value, so it would be generated and passed on to this axon terminal here. And then again, because there is a gap between these two, the axon terminal here and the dendrite here, so that which is known as the synaptic cleft. So the same way, the neurotransmitter will be released, right, which causes action potential again through the opening of the chemical gated sodium ion channel, which causes depolarization, right? So this is actually how synapses are a little different here. Like the action potentials generated here are different as compared to what they are generated here in the myelinated sheets. Got it? Okay, so this is just a view of terminal axon here. So this is what you see. This is axon, this is its terminal part, right? And then this is another neuron. So this is the dendrite now. See, so this is the axon of one neuron, and it is actually right next to this dendrite here, right? So this is what is actually the space, what we call as a synaptic cleft, right? So there's a little gap between them. They're not exactly joined together. So there's a small gap, which is around 20 nanometers in size, right? So when these two neurons meet, like, so you can call it as neuron 1, this is neuron 2. So when they meet, they usually do not touch, right? So this is a gap between them, which we call a synaptic cleft, right? So this is actually these three things together. So this part of the axon, right? So which we will call as presynaptic membrane, right? This synaptic cleft, this gap between them, and then this one for the second neuron, this will be what we will call as postsynaptic membrane, right? After the synapse, right? Postsynaptic membrane. So these three, these together, so the part of these two neurons near to the cleft plus the cleft, this is what we call as a synapse. Clear? Okay, then here like a few structures are also visible. So you can see this is the mitochondria. So again, like for production of ATP, like these are the vesicles actually that contain the neurotransmitters. And these vesicles are found only in this neuron here, which is the presynaptic part, right? So it will be found in the axon here. So these vesicles actually contain these neurotransmitters, right? And now if you look at the postsynaptic membrane, these are the ones that carry the receptors for these neurotransmitters. So as these neurotransmitters are released through exocytosis, they get attached to the respective receptors, right? And then if the impulse is high enough, they will actually lead the sodium channels to open. They would diffuse in and they would actually depolarize this part of the membrane. And then this would lead to the generation of action potential, right? But if the stimulus is not high enough, the nerve impulse will not be transferred. Okay. So let's talk about this mechanism of synaptic transmission in detail now. So these impulses, as we just discussed, they cannot jump across the synapse, right? So instead of that, these molecules of the transmitter substance or a neurotransmitter, they're released to stimulate the next neuron, right? So just like in a myelinated axon, as those nerve impulses were jumping across the nodes of Ranvier, that is not going to happen across the synapse, right? So here, this neurotransmitter is actually will do the job. So it will be released, and this neurotransmitter will actually stimulate the next neuron, right? So this is a basic outline of what events occur, and we'll talk about these events in detail. But the basic events are an action potential occurs at the cell surface membrane of the first neuron. So the first neuron is what we call as a presynaptic neuron. So if you look at this part here, right? So an action potential will be generated here, right? At the presynaptic membrane. Okay. So as this action potential generates, this causes the release of transmitter substance into the cleft, right? So as action potential will be generated, this will cause the release of this neurotransmitter. So how this all happens, we will talk about the detail also, but these are the basic gist of what happens. So an action potential is generated, which causes these vesicles to release these neurotransmitters, right? The molecules of the transmitter substance then diffuse across the cleft and bind temporarily to the receptors of the postsynaptic neuron, so which you're looking at here. So like see, this is the neurotransmitter that is connected or attached to this receptor here. So it binds to it temporarily, right? Now the postsynaptic neuron responds to all the impulses arriving at any one time by depolarizing, right? So this attachment of neurotransmitter to the receptor, right, this will actually lead to depolarization as sodium channels would open, right? And if the overall depolarization is above the threshold, only then it will send a nerve impulse. See, so these are the events that happen at synaptic transmission. Okay, now so let's talk about them in detail, and as per our CI specifications, we have to talk about cholinergic synapse, right? So this is the detailed events that happen. So what happens first? The cytoplasm of the presynaptic neuron, remember the first one for the axon, it contains vesicles of transmitter substance, so as we just saw in the previous diagram. And now there are around 40 different transmitter substances which have already been known, right? So there are noradrenaline and acetylcholine. So acetylcholine as an abbreviation is written as capital A, capital C, small h, right? So noradrenaline and acetylcholine are found throughout the nervous system, right? So these are neurotransmitters, whereas others such as dopamine, glutamic acid, and gamma-aminobutyric acid, which are in short known as GABA, they occur only in the brain. So around 40 are known. These are just few examples. And out of these, we are talking about acetylcholine, right? This is what we are going to talk about. And this is actually what will lead to this cholinergic synapse. Okay, now, so the synapses that use acetylcholine as a transmitter are known as cholinergic synapses, right? So since the neurotransmitter involved in this case is acetylcholine, we will call them cholinergic synapses. Okay then. So as an action potential occurs at one place at the axon, local circuit depolarizes the next piece of the membrane. So this is what related to the action potential that we have just discussed in the previous part of the lesson, right? So this local circuit depolarizes the next part of the membrane, right? It stimulates the opening of voltage-gated sodium ion channels, right? It was causing the influx of sodium ions, and this actually propagates the action potential, right? So this is all what is happening at this part here right now, because action potential was actually being generated, right? And through this part, as action potential is generated, voltage-gated sodium ions channels these open, sodium gets in, right? So this part of the membrane becomes depolarized. Now, what is different in this part of the membrane is that now the part of the membrane of the presynaptic neuron that is next to the synaptic cleft, the arrival of action potential as it reaches that place, it also causes the voltage gated calcium channels to open, right? So this is what is different here. So as those voltage gated sodium channels open, voltage gated calcium channels would also open, right? At this part here, as action potential reaches here. So along with voltage gated sodium channels, voltage gated calcium channels would also open, and through that calcium would actually move inside. Okay, so this action potential causes not only the sodium but also the calcium ions to diffuse in the cytoplasm of the presynaptic neuron. Now the thing is, in that area, there are virtually no calcium ions at all. In the cytoplasm, there are no calcium ions, but what happens, because there was a lot of sodium there, there's a lot of calcium there in the tissue fluid surrounding the synapse. So this means they actually enter through a steep electrochemical gradient because there is a lot of calcium outside, right? So it enters because of a steep concentration gradient. And then again, because of this difference in polarity, so because of this electrochemical gradient, calcium ions also enter. So this is this influx of calcium ions actually, which stimulates these vesicles, which contain the neurotransmitter acetylcholine, and these vesicles start to move towards the presynaptic membrane until they fuse with it, right? And then through exocytosis, they empty their contents into the synaptic cleft. So this happens because of the calcium, right? Each action potential causes just a few vesicles to do this. So all of the vesicles do not fuse at once, right? So with every action potential, only few vesicles do this, but each vesicle can contain around 10,000 molecules of acetylcholine, right? Now the acetylcholine diffuses across the synaptic cleft, and usually it happens in less than 0.5 milliseconds. So this is how fast this whole mechanism is. See, so just as a review, so as the calcium goes in, it causes these vesicles, which are already inside this part, right? It causes them to come near this presynaptic membrane and fuse with it. And through exocytosis, it releases the neurotransmitters into this area. Okay, now when we come to the cell surface membrane of the postsynaptic neuron, it contains receptor proteins, right? Now these receptor proteins, a little part of it contains complementary shape to the part of acetylcholine, right? Not all of it. So part of this receptor protein has a complementary shape to the part of acetylcholine molecule. So that's the reason acetylcholine can temporarily bind with the receptors. So because of that, as this neurotransmitter attaches to the receptor protein, this causes the shape of the protein to change. Because of that, the sodium ion channels would open, right? So as the sodium ion channels open, they diffuse into the cytoplasm of the postsynaptic neuron and hence they depolarize the membrane. Okay, now the thing is these channels are actually chemically gated, right? These are not voltage gated as we have already discussed there in the axons, right? They're not voltage gated, they're chemically gated because these sodium channels are actually opening because of the neurotransmitter getting attached to the receptor. And as the receptor protein, it changes its shape, only that allows the potassium channels, the sodium channels to open, right? So that's the reason the sodium ion, these channels are actually chemically gated. They are not voltage gated, right? So as they're stimulated by these chemicals, neurotransmitters, and not by voltage change. Now, if the acetylcholine remains bound to the postsynaptic receptors, the sodium channels would remain open. So remember we just discussed that actually they temporarily bind with the receptors, right? So if they remain attached, these sodium channels would remain open, right? And this postsynaptic neuron would actually be permanently depolarized, right? So this is where the acetylcholine will actually be recycled to prevent this from happening, right? And definitely because the neurotransmitter will actually be wasted because of that, right? So there is an enzyme which actually breaks down this acetylcholine, right? So this is what is happening at the postsynaptic membrane. The neurotransmitter gets attached to the receptor, right? Which has a little part of it which is complementary. as the neurotransmitter binds to the receptor, the shape of the receptor protein changes, which allows sodium ion channels to open. They diffuse in and they depolarize the postsynaptic membrane. And remember, this entry of sodium ions is through the chemically gated ion channels, not through the voltage gated channels, right? Okay then. Now, this synaptic cleft contains an enzyme, and the name of the enzyme is acetylcholinesterase. This enzyme, which catalyzes the hydrolysis of each acetylcholine molecule into acetate and choline, because remember, it has to be recycled. So the enzyme that is used is acetylcholinesterase, which is found in the synaptic cleft, right? It catalyzes the hydrolysis of each acetylcholine molecule into two parts, acetate and choline. Now, this choline is taken back into the presynaptic neuron. It goes back, right? And then it is combined with acetyl coenzyme A, and hence it produces acetylcholine once more, right? The acetylcholine is then transported into the presynaptic vesicles and ready for the next action potential. So this is how it is recycled. It goes back into the vesicles, right? So in the enzyme acetylcholinesterase catalyzes the hydrolysis of acetylcholine into acetate and choline. Choline is taken back into the presynaptic neuron, where it combines with acetyl coenzyme A and it forms acetylcholine again. This acetylcholine is transported into the presynaptic vesicles and then it becomes ready for the next action potential. So this entire sequence of events from the initial arrival of action potential, through which though that calcium, the voltage gated calcium channels also open, which cause those vesicles to fuse with the presynaptic membrane, and through exocytosis the neurotransmitter was released. Till the time the acetylcholine is actually broken down and reformed, this whole process takes around 5 to 10 milliseconds. The depolarization of postsynaptic neuron only leads to the generation of an action potential if the potential difference is above the threshold for that neuron. So as we have already discussed, it will not be generated if the threshold is below, right? So if it is below the threshold, no action potential will be generated at all. So again, remember the all or none principle, right? The chance that an action potential is generated and an impulse is sent in the postsynaptic neuron is increased if more than one presynaptic neuron releases acetylcholine at the same time or over a short period of time, right? So this is how the chance is increased. Like if the more acetylcholine, actually the more neurotransmitters are released, right? Or they're released in short span of time. So this is actually the whole process of cholinergic synapse. Okay, so just have a look at the diagrammatic version as well. This is the axon terminal. This is a presynaptic membrane, right? And this is the part of the next neuron, which is dendrite. See, and in between is the synaptic cleft. So you can see this part of the axon. This is the part which contains mitochondria. It contains these vesicles. So these vesicles are only present here. So again, if you talk about one direction of passage of this nerve impulse, so this is the reason also, because these vesicles, these neurotransmitters are only present in this axon here, the presynaptic membrane, right? And only through these, they're actually, through exocytosis, they're actually diffused into the synaptic cleft, and through the synaptic cleft, they get attached to the receptor. So it's again a one-way process, right? So you can see these voltage gated sodium channels open, right? And because of that, these voltage gated calcium channels also open. Now there is no calcium here in the cytoplasm, but since this calcium enters, it is this calcium which causes these vesicles to fuse with the presynaptic membrane, release the neurotransmitters, which get attached to the respective receptors, right? The receptors change their position and their shape, and then they allow these chemical gated sodium channels to open, right? And cause depolarization here. Clear? Okay, so this is just a brief summary. So this is how where the action potential arrives. So this is the initial phase. Action potential arrives. Action potential stimulates the opening of voltage gated channels of calcium ions, which diffuse into the cytoplasm. So you can see the cytoplasm is getting in, right? The calcium ions cause these vesicles containing acetylcholine to move towards the presynaptic membrane and fuse with it, right? It is then released across the synaptic cleft. Then acetylcholine molecules bind with the receptors, right, at the postsynaptic membrane, causing them to open the sodium channels. So you see, as they're being diffused, Here in the synaptic cleft, they cause these chemical gated sodium channels to open, right? And causes depolarization, right? So this is the scenario of how this neurotransmitter is released and how it brings about depolarization at the postsynaptic membrane. So much of the research on synapses actually has been done at synapses between a motor neuron and a muscle, right? So it is not actually being done between two neurons. So most of the research has been done between a motor neuron and a muscle. So the motor neuron forms a motor end plate, right? And the effector, the muscle fiber actually, and the synapse, that's why it is known as a neuromuscular junction, right? Because the research has been done at these places only, right? Most of the research. So such synapses function in the same way as described earlier. An action potential is produced in the muscle fiber, which may cause it to contract. So we will discuss this about when we talk about the contraction of skeletal muscles in the next lesson. But a neuromuscular junction is what? When a motor end neuron forms a motor end plate with each muscle fiber, and the synapse is called as a neuromuscular junction. So this is another diagrammatic view of how the whole process starts. So you may see this is the presynaptic neuron that they've shown here, right? These are the vesicles in the presynaptic neuron. These are the receptors in the postsynaptic neuron, right? And all the steps then, so have a look, right? And finally, the neurotransmitters, they're recycled or they're destroyed once the impulse is sent. So that's the whole process. So just have a look at all the steps. Okay, so let's talk about the role of synapse. So what's the purpose? So synapses ensure one-way transmission. Impulses can only pass in one direction at the synapse. So remember the part, since the vesicles are only present in the presynaptic membrane, right? It's only present there. So synapses allow integration of impulses. Each sensory neuron has many branches at end of its axon that form synapses with many intermediate neurons. So see this part? This is where this integration will take place, right? Because there are a number of branches here, and each branch actually is connected with a number of dendrites here also. So this actually involves a lot of integration here. Now synapses also allow interconnection of nerve pathways, right? So this happens in two ways. Individual sensory and relay neurons have axons that branch to form synapses, as we just saw in the diagram, with many different neurons, right? There are many neurons that terminate on each relay or motor neuron, as then they have many dendrites to give a large surface area for many synapses, right? So these are different roles, and synapses are also said to be involved in memory and learning, right? So these are four basic roles of synapses. So they ensure one-way transmission, right? They allow integration of impulses, right? They allow interconnection of nerve pathways. And finally, they are also involved in memory and learning. So let's talk about these advantages in detail now. So when we talk about one-way transmission of impulses, so impulses can pass only in one direction at the synapses because of the neurotransmitter. Like it is released only on one side and its receptors are on the other, right? So chemical transmission cannot occur in the opposite direction. So this prevents impulse from traveling the wrong way back to where they were initiated, right? So that's the explanation of one-way transmission. Now, when we talk about integration of impulses, each sensory neuron has many branches, right? That form synapses with many relay neurons. The cell body of each motor neuron is covered with a lot of like terminals of many relay neurons. Motor neurons only transmit impulses if the net effect of the relay neurons is above the threshold required to initiate the action potential. If the depolarization of the postsynaptic membrane does not reach the threshold, no impulse is sent. This stops the brain from being overloaded with sensory information. Impulses with low frequencies do not travel from sensory neurons to the brain at all, right? And when we talk about the interconnection of nerve pathways, individual sensory and relay neurons have axons that branch to form synapses with many different neurons. This means that information from one neuron can spread throughout the body to reach many relay neurons and effectors. There are several neurons that terminate on each relay and motor neuron as they possess many dendrites, right, which provide a large surface area for many synapses. This allows one neuron to integrate the information coming from many different neurons, right, and which is actually essential for decision making in the brain, right? So this slide is important. So have a read and understand the reasons that why these advantages and what is the explanation of these advantages of having synapses. Okay, so certain drugs also have effects of synapses, right? Now, there are drugs which are known as agonists, which actually acts as stimulants. They mimic the action of the neurotransmitters, right? And some that act against them are known as antagonists, right? So drugs which are agonists, they appear to have the same shape as the neurotransmitter, and they also behave like one of the receptors, right? So these activate more receptors. So these stimulants are mostly given to patients like with ADHD when you want them to focus, you want a response, right? So these are known as agonists because they mimic the same role of the neurotransmitter. Now, some drugs are called as antagonists. They block the receptors so they cannot be activated by the neurotransmitters, right? So this results in the activation of fewer receptors. So if you want like the responses to diminish, so in that case, antagonists are given. And certain drugs stop the action of enzymes which are responsible for breaking down the neurotransmitters. So we have different types of drugs which have different effects. So some have effects on enzymes, some have effects on the receptors, block them. So definitely the receptors will not respond to the neurotransmitters, and some actually behave like them. Okay, so there are some drugs which are known as amphetamines. Amphetamines are actually stimulants, right? They activate more receptors by stimulating the release of neurotransmitter from the presynaptic neuron, right? So these are actually used for like increasing the attention span. And there's some drugs like alcohol. What they do, they activate fewer receptors, right? So if they activate fewer receptors, so this will halt the release of neurotransmitters from the presynaptic neuron, and the responses would be delayed, right? Because if a nerve impulse is being passed quickly, so we'll have quick responses. But alcohol actually diminishes that because it activates fewer receptors, right? So these are certain effects that drugs can have on those receptors also. Okay, so finally we'll watch a video about synapses, and our topic about nervous coordination is done. In the next lesson, inshallah, we'll talk about the skeletal muscles, the details on how the action potential takes place there. So inshallah, see you then.
A neuron transmits nerve impulses through itself, but when it's time to send a message to another cell, most neurons will send chemicals to the next neuron across a very small gap between them. The junctions between neurons and the next cell are called synapses. There are three major types of cells that neurons will form synapses with: sensory receptor cells, other neurons, and effector cells. A sensory cell could be part of the eye, ear, nose, skin, or any place where senses are used. This sensory cell is connected to the skin and will pick up pain sensations. The pain message may be sent to another neuron that could be part of the spinal cord or brain. These intermediate neurons are also called interneurons. The message can then be sent to a cell that will have an effect on another cell, so they're called effector cells. In this example, the effector cell is going to signal a muscle cell, which would move this area of skin away from the source of pain. These three cell types together make a simple reflex arc, going from sensation to the central nervous system to an action. The chemicals released into the synapse are called neurotransmitters. Many neurons have a very small fluid-filled gap between the presynaptic cell and the postsynaptic cell. This gap is called the synaptic cleft, and it's only about 20 nanometers wide. It's a really, really tiny gap. Now that we know some of the basics, we can look at the steps of synaptic transmission in more detail. First, a nerve impulse in the presynaptic neuron reaches the end of the membrane. This causes calcium ions to diffuse into the cell through channels in the membrane. The influx of calcium causes vesicles with neurotransmitters in them to move and fuse to the cell membrane. The neurotransmitters are released into the synaptic cleft by exocytosis. They diffuse across the synaptic cleft and bind to receptors on the postsynaptic membrane. The binding of neurotransmitters triggers sodium ion channels to open and pass the threshold potential. The action potential is propagated along the postsynaptic neuron. Then the neurotransmitters are broken down and removed from the synaptic cleft. If there isn't enough neurotransmitter binding to receptors, then there won't be enough sodium flowing into the cell to reach threshold potential. This means that there is an all or nothing response. Either there is enough neurotransmitter to start the action potential and the impulse is sent, or there is no impulse. In this case, there was not enough neurotransmitter released to trigger an action potential, so the neurotransmitters will be broken down and the sodium-potassium pumps will restore resting potential in the postsynaptic cell. There are many different types of neurotransmitter, but we'll look at just one in more detail right now, acetylcholine. This neurotransmitter is made of an acetyl group and a choline, which must be combined together in the presynaptic cell before it's packaged into a vesicle. Then it can be released into the synaptic cleft. After acetylcholine binds with a receptor, it is rapidly broken down by acetylcholinesterase, an enzyme that's present in the synaptic cleft. The acetyl group and choline can then be reabsorbed by the presynaptic neuron and combined again into acetylcholine for reuse. One type of insecticide takes advantage of the acetylcholine receptors to paralyze and kill insects. Neonicotinoids are synthetic compounds that are similar to nicotine but bind permanently to the acetylcholine receptors. The acetylcholinesterase cannot break the neonicotinoids down, and the neurons can no longer receive messages or send action potentials. This is what leads to the paralysis and death of the insects. The effects are very small in humans and mammals, but the neonicotinoids don't discriminate between types of insects. Honeybees and other beneficial insects are inadvertently killed by neonicotinoids too.